Just one slide. So we discussed this point couple of times, but this is the basic central part of the molecular biology. That's why I tend to come to this part very much. Now we have learnt about proteins, what are proteins are, how do they function. We learnt about the nucleic acids, DNA, and here it is in RNA here. And in this picture, we can see actually three varieties of RNA. This is messenger RNA. These are the transfer RNAs, and these are the in this large and smaller subunits, we have a lot of ribosomal RNAs, rRNAs. So in this overall unit, we have three varieties of RNA itself. Among them, the most short-lived RNA, that means its half-life period is minimum, is the messenger RNA. So what you see here is there is a template, okay, and in the DNA itself. On the basis of the template, you get these chains of the RNA arranged, nucleotides in the RNA arranged, and you get an RNA. Similarly, on the basis of this RNA, three nucleotides together can get out, can code for one amino acid, and that's how a protein chain can be produced. So a messenger RNA of 300 nucleotide can code for, technically code for, I mean, roughly I'm saying, it's not accurate. Roughly it can code for protein of 100 amino acid, okay, or polypeptide of 100 amino acid. Now we have to minus one triplet code, that is the stop codon, in which the RNA, there is a stop signal. The coding, it's not, the translation doesn't wait until the full length of messenger RNA. Actually, there are three codons, which is, there is no tRNA to complement with. So these codons are called stop codons. That's called genetic codes. We'll discuss that in detail later. But just imagine this 300 nucleotide RNA can code for, or can give a signal to translate to have 100 amino acid polypeptide. And similarly, you just have to divide it by 3, the length of the RNA and messenger RNA to the protein. Now, what you see here, it's very important here, is that the production of RNA, they had a very nice device and all. When they are produced from DNA, very watchfully, they are exactly done through templates. You must have seen potters, when they make idols and all, they have a template on which they may put clay or plaster of Paris, and they will get exactly the same shape and structure of the idols of Ganeshji, idols of maybe Subhash Chandra Bose, whatever they want to make. So they do have templates, and these all systems work with a very specific templates. But are these three molecules are the main molecules of living systems? Yes, for processing and all, yes, these are the main molecules for hereditary information passing and execution of the information. But there are many, with these three molecules, you cannot complete life, okay? You can complete a virus. A virus is no life. Virus is a hacker of life, okay, if you remember that, okay? There are a lot of theories about viruses. Many people say that, viruses are loopholes of living system. So they did not appear before life appeared. No, they never appeared before life appeared. They actually appeared after life appeared, and there are some anomalies happened in living systems, and they tried to take advantage of it. Okay, so viruses are some faulty phenomena of living system. Often it is used for our benefit also. So we cannot completely blame on viruses. That increases our variability in our body, in the eukaryotes. So a virus can sustain with these things. Still, adenovirus or the large retroviruses, they often need a, lipid shell, lipid membrane, similar to that of our human cells or eukaryotic cells. So that means very small viruses like bacteriophages and herpes viruses, they can exist with protein and nucleic acid only, and they cannot live. Of course, they can infect with these two molecules, two varieties of molecules, but for them to sustain, they need a particularly functioning living cell, and particularly functioning living cells need other molecules as well. As I said, a membrane can be essential, and there could be another molecule, that is carbohydrates. Now let's see what can be a carbohydrate. Let's see, the central dogma we discussed. Okay, now you must be familiar with all these things. Okay, you love to wear cotton clothes. Your furnitures at your home, they are majorly wooden. Sugar, you add it to your milkshake, coffee, or your tea. And when you get into sports, often your PT teacher feeds this thing. This is a Glucon-D, right? So Glucon-D for instant energy. That's a very that's a trademark though, so trade name. It contains so all of these things are sugar. Okay, if you put all these things together, we are talking about, and here is a glue. Okay, and normal postal glues or very liquid glues, they are not synthetic, non-synthetic glues, are majorly made up of plant saps. And plant saps, you can say this babool plant, you must know that. They give out a glue. When you injure their bark, they release some gluey substance, and you can dissolve them. You can make your own glue bottle. Okay, non-synthetic glue bottle from gum arabic. Early days, chewing gums were also made up of glue. Okay, remember that. Okay, now they are synthetic. I mean, they are carbohydrate. Now they are synthetics. So you see, we are actually surrounded by varieties of things which are carbohydrates. Some are very sweet. Some are you don't know how it tastes, but it has got a very strong structural supports. Okay. And here some which can stick things. So in the regular central dogma, central principle of living system, did you see any carbohydrate operating? No, we did not see any carbohydrate operating. But for life, for a living cell to formation to pass, is just functioning of the cell. It needs carbohydrate to be present. Okay, so that is why carbohydrates are very essential. When you are feeling some bit of low, you can always take a bit of Glucon-D and you will always get recharged, because that gives maximum energy to your brain. In your body, whatever food you eat, it is finally, broken to glucose, hydrolyzed to glucose, to be more specific, I can say, and then glucose goes to your brain. In fact, 20% of the glucose you eat through different food is consumed by your brain. Okay, looks very small, right? But it takes almost 1/5 of your energy. Remember that. So let's understand what is a carbohydrate. Carbohydrates are actually, you must have learned about aldehydes and ketones, in organic chemistry. Remember that? So when you really see glucose, they are actually aldehydes. When you see fructose, there is this is fruit sugar. This is one of the this is the sweetest sugar available. I mean, naturally available. This is sugar. Okay, it's a ketone. What is the difference here? Can you see that? The carbonyl group here is at the tip. Okay, and at the carbon 1. Here the carbonyl group is at carbon 2. So why do we call carbohydrates? Because these are made up of carbon, and when you burn them, water comes out. So that's why they say carbon and water, they mix carbon. That's the most rough form of definition. But you see here, why water comes out, you see OH group, OH group, OH group, OH group. So many OH groups. How many OH groups? You can see that? In a 6, glucose is a hexose, meaning 6 carbon atom sugar. Okay, remember that. Glucose D we just discussed now is actually the glucose. It's a D glucose. We often call them dextrose. So it's rotation of polarized light. That's how we distinguish. There are 2 enantiomers. Like one is levorotatory glucose and another is dextrorotatory glucose. But dextrorotatory glucose isn't a common thing in normal. Naturally, you only see majorly see dextrorotatory glucose. Let's not bother about the rotation of polarized light. You must have learned that in your racemic mixture. You must have learned that in your +2, okay, in chemistry. I'm not going to see. See, there is one carbonyl group, and how many OH groups? 1, 2, 3, 4, 5. 5 OH groups. Okay. So there are 5 carbon, total 6 carbon atom. Only 1 carbon atom contains the what do you say, your carbonyl group, and others are majorly hydroxyls. Okay. So this is the reason, and each carbon has a hydroxyl and a H group. So when you normally burn this, what goes out? The hydrogen. If we all know that when you burn the sugar incompletely, bit of incompletely, you see a tar formation, dark colored tar formation. That dark colored tar is majorly carbon, and what goes out is actually the water. So HOH combines and they go out as water. This is a single carbohydrate molecule, and we normally call them a monosaccharide. Okay, we'll discuss monosaccharide, disaccharide, polysaccharide. Sucrose is a mixture of, or a dimer of, glucose and fructose. You see, and you see that is one is a aldehyde, another is a ketone. And sucrose, what is present in the cane sugar, is actually a dimer of glucose and fructose. In milk, we have galactose, you see, okay? And if you have glucose and galactose, you get lactose, that is disaccharide. What is the difference between glucose and galactose? Nothing. Chemically nothing, just you see OH group, one OH group, turns down, flips down, the purple group you see here. You see it towards the right side in case of glucose, and now it becomes left side in case of the galactose. Nothing much change here, but it's a different sugar now. Now, you would say that, what is this? Can't we call it enantiomers? Aren't they racemic mixtures now? No, racemic mixtures, for them to be different, there are other, what do you say, OH group, they play a role. Often the last or penultimate HOH group plays a role where the molecule can give completely oppositely rotates the plane polarized light. When you stock this molecule and make a solution out of it, the plane polarized light hits it. When it comes out, you will see the plane polarized light is rotated to some direction. Okay. So it can rotate right direction, it can rotate to left direction, clockwise or anti-clockwise. That's the idea. Okay. So now, is the hexose is the only sugar? No. There are varieties of sugar. Pentose we have already encountered, right? Pentose as in penta. Penta meaning 5. OSE meaning sugars are actually suffixed as OSE. Okay. So pentose. We have met, ribose. And the most important intermediate in photosynthetic pathway is ribulose. Okay, remember this, and we'll discuss it in the metabolic pathways. It is again another pentose sugar, but you can see it's a ketone here. We have glyceraldehyde. This is the intermediate in glucose breakdown. These are trioses, 3 carbon atom, trioses, 3 carbon atoms. Okay, glyceraldehyde. Why it is glyceraldehyde? It looks like glycerol, right? If you replace this carbonyl group with OH, it will be glycerol. Okay, but now this OH group is replaced with a carbonyl group. We call it a glyceraldehyde. Okay, so this is a major intermediate in glucose breakdown pathway. When we eat glucose, and this pathway is called Krebs cycle, we get ATP out of this glucose. These are the ways, these are the intermediates in that pathways. Then we can have dihydroxyacetone. This is another intermediate pathway, intermediate glucose. Here only the carbonyl group shifts to the middle here, okay, and this is a ketone. So when we have a aldehyde based sugar or glucose, what we call them? Aldose. Okay. What we call fructose, it's a ketone type of sugar. We call them ketose, right? What is the important thing here? They do have a carbonyl group. Okay. Either aldehyde, if it is at the tip, it becomes an aldehyde. If it is somewhere in the middle, it is a ketone. We all know that. This is the basic difference between aldehyde and ketone. So now chemically we understand carbohydrates are like this. These are the monomers. Now see, the point here is why we see this galactose with a single flip of OH group, it becomes a different sugar. The reason here is this. Normally, when we write and represent sugars, we normally write as a formula or in this linear manner. Okay, this is the linear manner. Carbon number 1, 2, 3, 4, 5, 6. Okay, so if you see this, I think this is visible. So yes, 1, 2, 6. This is visible most likely. So here you see here, the problem here is that in the real life or in naturally, when you take the sugar and allow it to settle, you just make a linear chain of sugar. Just make a linear. If you allow it to settle, what it does is it's a reversible reaction, though. Okay, what it does it makes a ring out of it. What is this ring? Does it end and closes with the complete carbon? No, it doesn't close with a carbon. From carbon 1 to carbon 6, it doesn't make a closed ring. What it actually makes, see what it actually makes is carbon 1, the carbonyl group tries to react to the 5th carbon OH. Okay, so what will happen? This is a rearrangement reaction, nothing else happening here. Okay, no much H groups are lost. You see here, what is this? How does this happen? This O tries to connect to this carbon here, and now this carbonyl group is vanished. Okay, carbonyl group is vanished, and this carbonyl group will contain an OH, carbon 1. And now you see here, is any of the atom is left out? No, no atom is left out. What is overhanging out of this plate sort of thing? Okay, this is a hexagonal benzene ring sort of thing. OH and H groups are dangling outside. Okay, and there is at the 6th carbon atom, which also contains an OH group, also dangles outward. Okay, it stands outside the plane of the overall ring. This is a very acceptable structure or energetically favorable structure. Yes, this ring structure is highly energetically favorable structure for any kind of carbohydrate, especially hexose and pentoses. Okay. Trioses, we are not very much sure. Some people say it is, what do you say, ring, and some people say these are linear trioses when we are discussing about glyceraldehyde. So this is how your glucose work. It makes a ring. Often this ring, makes a chair-like structure, or it looks like a boat-like structure, so that the 4th carbon atom, and 1st carbon atom, they actually bend upward. So that depends, what about your fructose? Fructose is a different molecule now. Fructose, because the carbonyl group is present in the 2nd carbon atom, you just have to see that fructose will have 2 dangling carbon atoms, and it will make a 5-membered ring. Okay, this is a 6-membered ring. This will have a 5-membered ring. Okay, this is how fructose makes. So in a sugar, in sucrose, you have actually a glucose 6-membered ring attached to a 5-membered ring, that is a fructose. Now, what is this happening here? This is when monosaccharide of these glucoses, 2 monosaccharides or 2 monomers of glucose, trying to link to each other to form a glycoside bond. What is this glycoside bond? These are the bonds which contains an oxygen. It's kind of an ether bond. Okay, where oxygen is intermediate, is between the rings, both of the rings connect to an oxygen molecule, oxygen atom, and then this bond is stabilized. Okay, so how does this glycoside linkage happens? See, this carbon 1 of one molecule reacts with carbon 4. Here is the carbon 5, here is the carbon 6, remember. Huh? This is carbon 4. And here the OH group reacts with each other, or we can say a condensation reaction, generating a water molecule. Okay. Now, if you want to break this glycoside bond, similarly, a water molecule has to be consumed here, and same OH group will be regenerated here. If you break it, we call this process hydrolysis. Okay, so you can generate monomers of that from this glycoside linkage. What happens in case of glucose and fructose? Same linkage happens. Here you see 1 and 4 carbon atoms are involved, so we call it 1,4 glycoside linkage. Here carbon 1 is involved of glucose, and which carbon of fructose is involved? Looks like it is 1, 2, 3, 4, but it is carbon 2. Is the second carbon of fructose involved. Why it is carbon 2? Fructose is actually a inverted enantiomer of, and it's a levulose. We can call it a levulose because its orientation of its hydrogen hydroxyl groups are mirror opposite to that of the glucose. So it's called levulose. So that's why, you know, sucrose faces fructose faces its face towards the face of glucose. Here they are running back to back like a train. In case of maltose, when glucose fuses to glucose, in case of maltose formation, they make a train sort of thing. Here they face each other. You can see that here, fructose and glucose. Okay. So this is how disaccharides are formed, and these are the reactions which is happening. Okay. So glycoside linkage is formed, and what is the reaction? Dehydration or condensation reaction, you can say. Okay. Now let's discuss about different forms of carbohydrates. Monosaccharides we just discussed, glucose, fructose, galactose we just discussed. Disaccharides, sucrose, lactose is a disaccharide, glucose and galactose. Maltose, glucose plus glucose is maltose. Sucrose is glucose plus fructose is sucrose. When I say disaccharide, meaning 2 sugar molecule, when you hydrolyze them, okay, then you get 2 sugar molecules. Monosaccharide, you cannot hydrolyze them further. Okay, they are already 1 sugar molecule. If you treat them harshly, they become carbon or maybe smaller molecules. Then we have oligosaccharides, often trisaccharides, and 10 sugar molecules also called. This is a you know, bit of what do you say, fishy. Many people define oligosaccharide different way. This table says 10 saccharide. Until 10 sugar molecule, we call it oligosaccharide. Raffinose is one example. We have polysaccharide. What are the polysaccharides we know? Starch, glycogen, cellulose. What are the starch we use? Starch is there in your rice. The sugar you eat from your rice is actually starch, or you take it from your wheat. Okay. If you are a meat lover, you get this variety of. Many people, you know, who are very much they love non-veg so much. They feel really good once they eat a meal of mutton or chicken. Actually, what makes them feel immediately good is not the protein. It's the glycogen what feels them good immediately. Okay, this is the reason why your liver and and muscles look red. Not only RBCs, okay. Red meat contains more of glycogen. That is why when you boil the red meat, they do not turn white. In case of chicken, it turns white, but mutton, goat meat, they do not turn. They turn brownish. Okay, this is because of presence of glycogen. This is a colored. It gives out some color. Starch doesn't give much of color. It is white. Cellulose, we know it is whitish in color. So we just understood a basic differences of carbohydrates. Now we try to understand what are the polysaccharides. Okay, so we just discussed polysaccharides are there, and we saw cotton. We use furniture. Basic structure of your plant systems. Take its cell wall. From cell wall, it starts. The structure building starts. Right from that stage, you get these basic structures. All are made of carbohydrates. Consider redwood tree. Consider bamboos. Consider the coconut tree, which can stand supercyclones. Their all structures are made of carbohydrates, and the major carbohydrate which makes it is a polysaccharide, that's a cellulose. Okay, now you see this how this made. You can see this is the cell wall, and cell wall contains a lot of microfibrils. These microfibrils, very thin, thin, often called nanofibrils. They are often 80 molecules long, and you can see that each microfibrils, when you zoom it out, you can really see these individual carbohydrate molecules of polymers of beta monomer of glucose is there. Same glucose, but here you get so much strength without eating anything. You cannot. Now, in case of insects also, you see in shrimps and crabs, those who are fond of seafood, they love crab probably, and crushing the claw of a crab is very difficult with your teeth because of this presence of chitin. This is another molecule. It's again a carbohydrate, long carbohydrate chains, polymer. But here you have this carbohydrate, their monomer do have a special feature. Can you see this? Amine, okay. Okay, so here is a cicada moth. It's the largest moth, one of the largest moth coming out of its molding. It's actually shedding its exoskeleton. This is the old exoskeleton, which all contains chitin. Okay, cockroach, crabs, they all have their exoskeleton made up of chitin, and that's a very strong polymer. Often we use this chitin for surgical, as a surgical thread. Often we use silk. Silk is a protein though, okay, and sometimes we use chitin for the same purpose. So we understand now structural support. Carbohydrate can give us very strong structural support, and majorly animals like insects. Actually, that is the largest volume of animals present on the surface of Earth. Those are insects, and of course plants. They have their structural support, which is made of carbohydrate. So no fuss, no coding, just linkages. For this chain linkage, do you think there is a coding or templating system? No, nothing like that. It simply grows like a crystal growth, kryptonite crystal growth. So here is the carbohydrate, you see, and sorry. Now we discussed about another which are relevant to food, starch and glycogen. These are the carbohydrates which are more relevant to our as a food. Starch, we get it in potatoes, starch appears as granules. So food relevant carbohydrates often appear as granules, not fibers. Okay, glycogen also appears as granules. Often glycogen develops in spirals. You can see that. Okay. Starch is majorly, when plant stores food, it makes structures out of half of the 99% of the glucose. Some glucose is stored for food purpose, for getting energy, and that is in the form of starch. Animals, they want to save glucose for unusual time, burst release, when people work out more and all. They cannot depend on each time eating food and all. So that's why they store glycogen locally near the muscles, often in liver, to get sugar immediately and process it to make ATP there. So that is how, so for storing food or storing glucose in a small area, they often use to make it granules, and in case of plant, they store in starch, and in case of animals, they store in the form of glycogen. There is a difference in the bonding pattern. But here you see, there is another important question, is that a cow can eat grass, they can digest grass, but you can't do that. Do leaf contain starch? Yes, a little bit of starch, but to get the starch majorly, you need to depend on the grains, food grains of fruit. Often some fruits contain starch. Stems contain starch, like sweet potatoes or your potato. They contain starch. Germans, they do not eat grains. They for starch or for sugar, they eat potatoes. Okay, Britishers do the same. So we can digest those starch, but same starch is a polymer of glucose, and cellulose is a polymer of glucose. We cannot digest starch, but cow can do. What is the point? When you see maltose, glucose linked to 1,4 linkage. You can see this here. We saw in maltose, right? So when you attach multiple maltoses, like this glucose disaccharides, on, that becomes your starch. And these are 1,4 linkages. This first carbon atom links to the fourth carbon atom. Both the OH groups club together, leaves the oxygen and throws out a water molecule, and that's all. Your starch is done. Starch is getting ready, and gradually individual glucose molecule is being added. Why can't we digest cellulose? What is cellulose here? Yes, it is 1,4 linkages here. Okay, same 1,4 linkage, but there is a bit of difference here. Here it is alpha glucose monomer, and here it is a beta glucose monomer. What is this beta glucose? Okay, you see here, the OH group here in the first carbon atom is downside in the alpha glucose, and in beta glucose it is upside. Okay, so the bonding will happen, rather, you can see that this bonding happened downside because the OH group is down. Here, the first carbon atom bond oxygen is upside, so the bond happens on the above portion. Same, it is in this fashion only, in the train fashion, but these bonds, the beta glucose linkages, 1,4 linkages, doesn't allow us to eat cellulose. We do not have any enzyme which can, our stomach never produces these enzymes to chop these kind of bonds. We have enzymes to chop this kind of bond. So as we just saw, that enzymes are very specific. For even a small subtle change in molecular structure, the enzyme may not work. We're very specific. So that's the reason we cannot digest cellulose, but a cow can. Okay. So that will be all on carbohydrates. There are certain, things, important roles of carbohydrates we will discuss in the next class. Thank you for your attention.
Thank you, sir.
Thank you, sir.
Bye.
Thank you, sir.
Thank you, sir.
Guys, take care.
Thank you, sir.
If you have
Thank
any
you, sir.
questions, you can write it in the comment box. I'll try to answer it. Yeah. Thank you.
Thank you, sir.
Thank you, sir.
Bye, students. Bye. Take care.